Hello students, I am teacher Svetlana, and in our today's lecture we will learn about the development of the endosperm and the development of the embryo, which is covered in the chapter Reproduction in Lower and Higher Plants. Development of the endosperm. The triploid primary endosperm nucleus repeatedly divides mitotically to form nutritive tissues called as the endosperm. So you have your primary endosperm, which is formed after the fertilization, which divides mitotically to form the endosperm. In post-fertilization changes within the ovule, the embryo and the endosperm are seen to develop simultaneously. But first we will have a look at the development of the endosperm, which will be followed by the development of the embryo. The other cells of the embryo sac disorganize sooner or later. Now, the formation of the triploid endosperm nucleus triggers cell division, which leads to the formation of the endosperm. Have a look at this image here. This is how our endosperm looks. Now, there are three different types of endosperm, that is nuclear endosperm, cellular endosperm, and herbuloid endosperm. Let us have a look at how each of these different types of endosperms develop. Students, the development of the endosperm, there are three types of endosperms which are decided on the basis of their mode of development. The first one is the nuclear type, the second is the cellular type, and the third one is the herbuloid type. Have a look at this image here. This first one is a nuclear endosperm, the second one is the cellular endosperm, and the third one is the herbuloid endosperm. Let's try to understand what is the difference between these three types of the endosperm. To start with the first one, that is the nuclear endosperm. It is the most common type and it is found in 161 angiospermic families. Here the primary endosperm nucleus repeatedly divides mitotically without wall formation to produce a large number of free nuclei. Have a look at this image here. You can see in the center here, in the center you have a large number of free nuclei. So first of all, you have a big central vacuole appears in the center of the cell, pushing the nucleus towards the periphery. Later, walls develop between the nuclei, hence multicellular endosperm is formed. But in several cases, cell wall formation remains incomplete. You have a look at this figure here from F to J. You will see first you have the one central your primary endospermic nuclei which divides into two. They get arranged and you have free nuclear divisions that is taking place. After a certain time, the cell wall develops around it. So this type of division is called as the nuclear division. Examples of plants showing nuclear endosperms is wheat, sunflower, and coconut. Coconut has multicellular endosperms in the outer part and free nuclear as well as vacuolated endosperm in the center. The next type of development of the endosperm that we will be seeing is the cellular endosperm. In some plants, division of the triploid primary endospermic nuclei is immediately followed by wall formation, so that the endosperm is cellular right from the beginning. It is mostly observed in 72 families of dicot members of the plants. So here you can see here that once the nucleus divide, immediately it is followed by the wall formation right from the beginning. Here have a look at this image here from A to E. These are the stages of cellular endospermic development. You have the nucleus of your primary endosperm that divides into two. Immediately there is a wall formation, and on the successive divisions, after every mitotic division, you have the cell wall that is formed immediately. So this type of division is called as cellular division. Then the third type of division is herbuloid type. Now this is the image of the herbuloid type endosperm. It occurs in the order of the herbuloids of the monocotyledons. In this case, the first division of the primary endosperm nucleus is followed by a transverse wall formation which divides the cell unequally. have a look at this image here from K to O. You can see here you have your primary endosperm nucleus. Now in this case, there is division of the nucleus, and the wall formation takes place, transverse wall, but the cells are divided unequally. The smaller end that you can see here is called as the chalazal end, and the larger end that you can see is called as the micropylar end, and the micropylar cell, and towards the chalazal end the cell is called as the chalazal cell. So it is divided into two unequal cells, chalazal cell and the a small chalazal cell and a larger micropylar cell. Then the nuclei in each cell divides by free nuclear division, and then the walls develop between the nuclei in the micropylar chamber. So here now it continues as a free nuclear division. So first the division was like the cellular division, then in the second case, in the proceeding further, it follows the free mitotic divisions of the nuclear and shows the nuclear mode of development. So this is called as an intermediate between the cellular and the nuclear type of the endosperm. So it shows both the characteristics. Initially it shows the characteristic of cellular endospermic development, and in the later stage it shows the characteristics of the nuclear endosperm development. Students, we have one more type of endosperm which is called as a mosaic endosperm. In this type, the endosperm containing tissues of two different types. This is called as a mosaic endosperm. So here in the mosaic endosperm, the endospermic tissues has two different types of tissues. Now in plants like corn, the endosperm contains patches of two different colors, and it forms a sort of a mosaic pattern. Have a look at this image here. You can see here you have two different patches in the endosperm. So two different colors appear, which looks like a mosaic pattern. So such type of an endosperm is called as a mosaic endosperm. Students, now let us have a look at the development of the embryo. The process of the development of the zygote into an embryo is called as embryogenesis. So obviously this process happens after the fertilization. So as a post-fertilization processes, we have the development of the endosperm, which we just saw, and next part is the development of the embryo, which is called as embryogenesis. The embryo is developed at the micropylar end of the embryo sac. Now, the development of the embryo triggers only after certain amount of endosperm is formed. After fertilization, the embryonic development begins. So have a look at this image here. This is showing you stepwise the different stages in the development of the embryo, and here you can have a look at the mature embryo, which has two cotyledons, means it shows it's a dicot plant, and then it has an outer seed coat. So we will have a look at these stages. Students, have a look at this images here. This is showing you the dicot development of the embryo in dicot plants. This is monocot development of the embryo in monocot plants. So let us have a look at the dicot plant development of the embryo here. The first image shows you the zygote, which divides to form a two-celled proembryo. So this is the zygote, your A. Then it divides to give you the two-cell proembryo. The larger cell, the upper cell towards the micropylar, is called as the suspensor initial cell, and the smaller cell towards the chalazal end or the terminal is called as the embryonal initial cell. Students, the suspensor cells here, they undergo division, which is a transverse division in one plane, to produce a filamentous suspensors, which is around 6 to 10 cells long. The first cell of the suspensor towards the micropylar end becomes swollen and functions as a hostorium or a hostorial cell. This is a hostorium or a hostorial cell. The lowermost part of the suspensor, this one, is called as the hypophysis. Now the suspensor helps in pushing the embryo into the endosperm. The embryonal initial undergoes 3 successive mitotic divisions to form an octate. So if you have a look here, you can see the octate here. The plane of the division is at right angles to each other. The lower tier of the octate gives rise to hypocotyl and radicle, whereas the 4 cells of the upper tier forms the plumule and the 1 or 2 cotyledons. The hypophysis further divides to give rise to the part of radicle and root cap. So in this image here, you can see the root cap, which is along with the radicle. Now subsequently, the cells of the upper tier of the octate divide in several planes so as to become a heart-shaped structure. If you look at this figure here, you can see the heart-shaped structure. Now this heart-shaped structure forms the 2 lateral cotyledons at the terminal plumules. Further enlargement of the hypocotyl and cotyledons results in curvatures in the embryo, and it appears like a horseshoe shape. So you can see the radicle here and the plumule here. So till here, the development of the embryo is similar in the dicot and the monocot plants. The differences appear later. If you look at the monocot embryonic development, in the monocot embryo, single cotyledon will occupy the terminal position, and the plumule is present in the lateral position. Now the single sheath of shaped cotyledon is called as a scutellum. If you see this is your scutellum. The protective sheet of the plumule is called as colleptile. This is the protective sheath on your plumule, which are called as colleptiles, and that of the radicle is called as coleorhizae. So this is the protective covering on the radicle, which is called as coleorhizae. Finally, the ovule is transformed into seed and the ovary is transformed into fruit. So this is how the development of the embryo takes place. Students, have a look at this image here. These are the real-time microscopic images of the development of the embryo, and as said, finally the ovule is transformed into the seed and the ovary into the fruit. So if you see the end product here, you can see the outer seed coat that gets formed, which is covering the developed embryo. So this is how the development of the embryo takes place. Thank you, students, for participating in this lecture. We will meet soon again with the next topic. Till then, take care, be safe, and keep learning.